Hello and welcome to Jason Live. My name is Patrick Shea. We're back once again with our STEM career series where we learn about careers in science, technology, engineering, and math from role models working in those fields. Today's role model is Denise Akob. Denise is an environmental microbiologist with the U.S. Geological Survey. Denise studies how environmental microbes impact energy production, clean up contaminants, and do lots of other cool things you would not normally think of when you think of microbes. We'll connect with Denise to learn about her research and career path in just a moment. But before we do, I just want to remind everybody out there that today's event is live and interactive. We'll be taking your questions, and we hope you participate in our polls. To do both of those things, just use the box right below this video window. We'll try to get as many of you involved in today's program as possible. Right now, it's time to get Denise involved in the program. Welcome, Denise. Thanks for joining us today.
Thanks for having me today. It's good to be here.
All right, so let's start with the basics. We certainly want to know what it is that you do. And I know when we talked before, there was some discussion about what the best term for your job is, whether you're an environmental microbiologist or a biogeochemist. So talk to us a little bit about that. But also, I think, just for the benefit of everybody out there, to lay the foundation here, we're going to be talking about microbes. Can you just tell us what a microbe is?
Sure. So microbes are a lot of different organisms, actually. So they can be anything from, there's two different kinds of single-celled organisms that I study. Those are bacteria and archaea. Those are called prokaryotes. So those are single-celled organisms that don't have a nucleus. Bacteria include the kind of microbes that will make us sick, so the germs that many people know about. But what I study are actually the microbes that really help the environment and help keep the earth clean and healthy and things like that. Other kinds of microbes can include fungus, so fungi. The organisms that even make mushrooms are microbes for part of their life. And there's also things called multicellular microbes, so things like diatoms or algae, if you've ever heard of those. But again, I'm working on the organisms called prokaryotes, and those are the single-celled organisms. And they're really distinguished from microbial eukaryotes by the fact that they just have a cell wall and they don't have any nucleus inside. So if you guys have learned about that in biology, then you know what the difference is.
All right, we've been getting a ton of questions in already, and I do want to have you talk about what your job is and what your research is. But we've got a comment here from Joel, and Joel is a teacher, and Joel says, having taught microbiology in high school before, I always teach about microbes in my general bio classes. In the book Missing Microbe, Martin Blaser states that many of our everyday chronic epidemics are caused by trying to be too clean. I'm a strong believer in getting kids outside, so I hope you mention the value of microbes and also the wonders that are being found out in our environment. What do you have to say about that, and how does
Well,
that
Joel, I
factor
really
into
appreciate your
your?
comment. So I'm a mom. I have a three-year-old, and my husband and I believe strongly in this idea of, that chronic epidemics can cause sickness, and especially because people are too clean. And we like for our daughter to get dirty and play around in the dirt because we think that her being involved in that natural environment from a lot of these studies are really good for her overall health. One of the things that really ties to the work that I do as an environmental microbiologist, linking to what you're talking about, is that microbes don't live by themselves. Just like most humans don't live by themselves. We live in a community of people. Your school is a community, your classroom is a community. So if you see the picture here in the bottom kind of in center, you'll see a whole bunch of different funny shapes. Those are all different kinds of microbes that are living in a community. And the microbes all interact with each other or have microbe interactions. A lot of the germs that will make us sick are because sometimes if we wash our hands too many times or use really strong antibacterial soaps, it'll kill off some of the microbes that are in that community, leaving maybe the nasty bugs, the ones that can make us sick, behind. And if those organisms then become kind of an individual in a community and no longer a part of a community or by themselves, then those are the organisms that can get really, really abundant and make us sick. So it's often really important to have communities of microbes that can then kind of work together to kind of keep your environment healthy, be it your environment outside in the soil or even the environment that's on our body. So I guess with that, I think it might be a good time to really define what I do as an environmental microbiologist. Pat, do you think you can put that other picture up again of what an environmental microbiologist is?
Yeah, why don't you start telling us about it? We'll dig
Sounds
that up
great.
for
Good.
you.
I just wanted to use that as an image. So I talked about how we have a microbial community. So I study microbial communities in the environment. Now, our bodies can be an environment, but I'm really looking at the natural environment outside of our bodies. So what occurs in soils, water, the oceans, even in rocks or sediments. And what I'm interested in understanding is what do the microbial communities look like in different environments? How do they interact with their environment? Maybe through controlling different parts of the carbon cycle. So a lot of people hear about greenhouse gas emissions and CO2 in the atmosphere. Well, microorganisms can control how much CO2 there is via their activity. So if organisms are decomposing dead plant or animal material, they can actually be putting CO2 into the atmosphere. Often we think of plants as being the organisms that actually fix carbon, but microbes can too. Microorganisms can also live on rocks and actually breathe in different minerals and cause minerals to dissolve or even to form. So I've actually studied microorganisms that make minerals. The image you see on the screen right now, you can see all those little green things are microbes. So those are really teeny tiny single-celled bacteria or prokaryotes, and they're living on an iron mineral. So this is actually rust. These organisms breathe rust. If they're found in the presence of oxygen, they'll actually die. So they have to live in places where there's no oxygen and can control the presence of minerals in the environment. Microbes are also really important for communicating with each other, and so that's another thing that we like to study in environmental microbiology. So
So these things are everywhere.
they're
They're
everywhere. Yeah,
everywhere, and there's
everywhere
so
from your
many of
belly
them.
button to deep rocks, 4, 5, 6 miles underground. They're everywhere.
So, I mean, can you give us a sense of, like, in terms of, I mean, numbers must be kind of really hard to get your head around, but like biomass, or how do they compare to, like, number of insects on the planet, which is also just a huge, huge number.
Yeah. So in 1 gram of soil, so if you imagine a gram is a very, very, very small amount. So I'm trying to think of what would be a good, okay, so here, think about a thing of chapstick. This is 4 grams, okay, this size. If you have 1/4 of this, there are over 10 to 100,000 different species of organisms. So imagine in 1 gram, you have 10,000 species of birds, for example. Granted, you can't fit them in that small, but that's how much diversity or how many different species are present in 1 gram of soil. And those organisms can be, billions and billions of cells in 1 gram of soil. And so the amount of carbon is thought to occur in the subsurface, so deep, deep underground from the soils all the way down to where you hit bedrock, is estimated to be the same amount of carbon kind of in all the plant biomass. So there, and that I would have to look up the numbers exactly, but you're talking about a lot of organisms in a very, very small amount of soil, for example. So these things are extremely abundant, and they make up a lot for the total carbon in our Earth's crust.
Yeah, those are really mind-boggling numbers.
Right.
And we've got a couple of related questions coming up now from Michelle and Yena. Have you ever found a new species of bacteria, and have you discovered any type of bacteria, new type of bacteria, or unicellular organisms?
Well, I was actually involved in the isolation and characterization of a new bacterium called Geobacter daltonii. This is an anaerobic bacteria. So one of the things that I work with primarily, my specialty, is working with organisms that can't breathe oxygen. So these are organisms that breathe other things other than oxygen. And one of our organisms, Geobacter daltonii, is actually isolated from a uranium contaminated site. And it can breathe uranium, and it can also breathe iron. And Geobacter can breathe iron, and the type of iron it breathes is actually rust. So if you ever see rust on a can or a nail, that's what this organism will actually breathe. And so it was really exciting to be involved in the isolation of this. It's a brand new species, and it's not very closely related to other organisms. So there's other species within the genus of Geobacter, but this is the first organism of its kind. What's been interesting since we started studying it, we found out that it had unique physiology. So the organism can actually degrade different hydrocarbons. So hydrocarbons are what are found in oil. So that's what makes up like oils and other kinds of like petroleum products. And so the organism is really interesting to think about how it might be acting in the environment and could be used for bioremediation, which are methods to clean up toxic waste using naturally occurring bacteria.
Well, we've got a ton of questions coming in here, some directly related to that, but the next two that we're going to tackle here are from Neha in Mr. SP's class. They're asking, to see bacteria, what power microscope lens do you use? And do the microbes freak out when you see them?
That's a fun question. So for what power of microscope, you can see them, usually large bacteria, and if they're stained with special stains that make them glow, you can see them maybe at a 40x power. Usually you really can't see them though until about 100x on a microscope. I've been really lucky though that I've been able to look at my microbes at about 40,000 times power, and that's using scanning electron microscopes or transmission electron microscopes. So these are very specialized microscopes, really big machines where they use electron beams to actually visualize the organisms.
Okay, we've got a couple of follow-up questions to what you were just discussing related to oil and gas. So Lisa's asking, what are the most common organisms, microorganisms, that turn coal into natural gas, and do they produce any harmful byproducts? And how much cleaner is natural gas than coal? And somewhat related here from Ophelia, we know that there are microbes that consume and break down crude oil. Can they be safely introduced to clean up spills like the BP Gulf spill without harming the environment later?
Good. So I'm going to answer part of those questions together. So one of the things is you're asking is whether or not the microbes can be introduced to the environment. Well, one of the things that I work on are organisms that are naturally occurring in the environment. So I'm interested in finding out who's present in an environment already and how we can promote their activity to make it occur more quickly. So in the case of a crude oil spill contaminated site, such as BP oil spill, or in this image you actually see this is me at a crude oil spill site in Bemidji, Minnesota, we're investigating microbes that are naturally occurring in the environment and that have become active because the oil spilled. And that's actually what happened at the BP oil spill site, is the organisms that ate all the oil were already there. Just the one that once the oil came in, the community, the population changed. So the organisms that were able to eat the oil became more dominant in the community, so their abundance increased. So there were more of them over time so they could be active. It's the same thing in the coal being turned into natural gas. We're looking at what organisms are present on the coal naturally and trying to understand how we can promote their activity. The organisms that primarily make natural gas, so converting coal to natural gas is actually a 3 to 4 step process. Microorganisms first have to break down the coal into small organic molecules that can be used by a second group of organisms, which can then be used by a third group of organisms, which include the methanogens. Methanogens are what actually make natural gas. They make methane, make the actual methane. These organisms are within an interesting domain of life called the archaea. They're single cellular prokaryotes, but they're archaea, so they have some really unique characteristics to their cell. The organisms within the archaea that do this include the genera Methanosarcina, Methanocida, and Methanomicrobiales, excuse me. I just had forgotten the name for a moment.
It was on the tip of my tongue, too.
On the tip of my tongue. So, but we're really interested in understanding who's really catalyzing those processes. Whether or not the coal we could have harmful byproducts is actually something we're studying right now. So it's an active part of our research, is to understand how, if we develop new technologies for enhancing natural gas production, will we also promote negative consequences to that? And that's something we need to find out. And so I'm working with some people, some toxicologists actually, who are looking to see if there's harmful byproducts. The other question you brought up was, is natural gas really cleaner than coal? It's really, I would say, a question that's up for debate. One of the main reasons that natural gas is cleaner than coal is because you're not producing NOx species. These are like nitrous oxides species, things that can contribute to greenhouse gas emissions. And also those are toxic byproducts that you really don't want to breathe as a human. Also, sulfur species can be produced. These are all things that are naturally present in coal, even mercury, and that when it's being burnt for combustion to make energy, it can be releasing a lot of nasty things to the atmosphere. And those are mostly the sulfur, mercury, and even nitrogen species that can be a problem. In terms of the amount of carbon dioxide or methane that could be released in the environment, I think they're about equal. It's just not nearly as many different things that can be bad for the environment.
Mm-hmm. We're going to jump to our next couple of questions next. These are from Sophie and Morgan. Have you ever encountered any species that gave you a hard time studying or just difficult? And Morgan wants to know, what's your favorite type of organism or bacteria?
Well, Sophie, I have that problem almost every day right now. So we have some organisms that are anaerobes, and they are breathing sulfate, and so that's a compound that's common. It's in rocks. It's often it's also very high concentrations in the oceans, and our organism just won't grow right now, and it specifically won't grow by itself. So we're trying to isolate an organism, sulfate-reducing bacteria, in out of a community. And one of the things is that it's really hard to work with these organisms under anaerobic conditions. This image you see is actually of an anaerobic chamber where we can work inside and under oxygen-free conditions, but it's really hard to manipulate the growth media, so the matrix that the organisms grow in under some of these conditions. You can have different chemical problems that will cause issues and make it really hard to select for the organism and really grow it well. What's my favorite kind of bacteria? My favorite organisms are actually the iron oxidizing bacteria. On my website, you can actually see a really cool picture of them. I forgot to get one for today, but you can grow these organisms on clear microbial media. And over time, they'll actually make rust. And that's how they gain their energy. And so you'll actually see the organism make these little streaks and dots of rust all over a clear medium. And so those are actually my favorite, just because I like the color change.
Yeah, I think that's actually the image that we used for your feature
Yes,
on
that's
top of the
me.
page. So yeah, that'll be back. Actually, you can go to the jason.org/live. It'll be there right now. We have time for just, I think, 3 more questions here as we're talking about your current career and your current work. This next one is from Amy and Alexa. They want to know, have you ever looked at something under a microscope that is dangerous?
Oh. Not that I know of, not on purpose. So
Okay.
maybe I saw it under the microscope and I didn't know it was dangerous. So most of the organisms that I culture are not known to be pathogens. So these are not organisms that would actually cause me to become sick. Now, I do work with some things that are dangerous. So you think you might see behind me a really fun radiation sign. I did most of my PhD work on uranium contaminated sites and looking at what kind of bacteria lived there.
Mm-hmm.
So we would often get samples that were uranium contaminated. I also worked at a heavy metal contaminated site due to uranium mining. And you didn't want to drink that water because it's really toxic. It could really make you sick. But the majority of the organisms I study are not dangerous. We're not growing them to be dangerous. So you can, as a microbiologist, you can make different cultivation media. So different ways that you culture a bacterium can change what organisms you might get into the laboratory. And so the media that I design isn't selective for pathogens. So I'm not trying to grow them.
So we've got time for just 2 more questions before we move on, and these are from Bobby, Amy, and Alexa. Do you find your job exciting, and if so, why? And what is your favorite part of having this job, and why?
Because I get to play. I get to play and do science. I love it. So it's exciting every day to be able to think of a new problem that you can investigate, just by playing in the lab. That's how it feels like fun to me. It always has. I often don't leave my work at work because I'm always thinking about how to solve a problem. And for me, it's always problem solving. So if we make an observation in the environment and say, oh, I'm going to go to a field site soon where we see in pictures that the stream is really orange and it looks like there's iron there, a lot of iron rust, and probably from contamination at the site. And one of the questions I could say is, well, are bacteria making the rust? Are these the iron oxidizing bacteria? And so I get to go play in the lab and see whether or not I can grow those organisms. We can use DNA to find out who the organisms are. And so I'm always getting to do very different things. I spend a lot of time writing right now in my office, but I also get to set up different experiments in the lab. I do things in using cool equipment like anaerobic chambers and gassing stations, and I get to look under the microscope sometimes. I get to see bacteria on plates, and other days I get to investigate their DNA. So it's just really exciting. And I also get to travel to cool places. I like that too.
It sounds exciting, and it just sounds like there's a lot involved. There's clearly a lot to learn from you about the work that you do and about microbiology in general. Unfortunately, we don't have time to get into more of that today, but we do want to learn, as we switch gears here to our next segment, about how you came to find this career. I mean, all of the things that you're talking about doing and all of the reasons why it's so fun and exciting for you now. Did you always know it was going to be like that? So we've got a couple of questions here that are going to get us talking about your career path. These questions are for Marissa, Amy, and Alexa. How old were you when you were first inspired to become a microbiologist? And who or what inspired you to get this job?
Well, that's interesting because I didn't really figure it out I wanted to be a microbiologist till I was probably about 23 years old. So I still was trying to figure out at that age what I wanted to be when I grew up. Um, I was always interested in science, though. I never realized it, but even as a middle schooler, I was interested in reading science books and learning about research. And I always kind of kept that going through high school, middle school, and into college. And when I was in college, I knew I wanted to go into science, but I didn't know what I could be when I grew up. So I thought I'd go to medical school. And I had some amazing professors who did research all the time on coastal environments. So they were marine biologists, but looking at the ecology of coastal environments. So, what kind of nutrient cycling is occurring. And they encouraged me to take a course called Tropical Marine Biology. And I got to go to Belize, Central America, and fell in love with doing research and being able to ask questions and go to cool places and use a lot of different methods to find answers. And so in college, I knew immediately I wanted to be a researcher. I wanted to be a research scientist. And my path took a couple turns because I did some internships and thought that I wanted to do like marine ecology. And when I went to graduate school, that's what I went to graduate school for. And I didn't realize that my interests were really in nutrient cycling. And nutrient cycling is catalyzed by microorganisms, by bacteria. And so I realized about a year and a half into graduate school that I wasn't really happy in the laboratory I chose. So I found a new PhD advisor to take me on. And that was in the oceanography department at Florida State University. And there was a project there doing strict microbiology of uranium contaminated sites. And I said, sure, I'll do anything. I just want to be a scientist. And I just fell in love with the study of biogeochemistry. That's really kind of what my degree was in as a PhD student. And doing that kind of work is microbiology. So understanding how the biology and the chemistry and even the geology of environments are linked, and understanding how microbes are responsible for making sure processes that are important are going on.
So was that a tough decision to make that switch at that point in your studies?
I was actually so unhappy in graduate school in the lab that I was in to start that I was going to either find a new position or I was going to leave science altogether.
Mm.
So it was a really hard time. I was very young. I was about 22, 23 years old in graduate school, and it was a really big change for me. And, I thought I would just go get a master's in this new lab. And within 6 months of being in the new lab of doing oceanography, I knew immediately that that's what I wanted to do for the rest of my life. And so I was really, really lucky that I found the right direction early. But it was a really tough time kind of finding what I wanted to do, if that makes sense.
All right, we've got some career path related questions that have come in now. This one's from Lori. What was your favorite book to read in middle school?
Oh my goodness, that's a long time ago.
You had
I
mentioned you like science books even then.
did, but what was it that I would read? Actually, I don't remember what book I liked to read in middle school, but this is a really unusual one. I had to write a paper in middle school and even high school, and early high school, And I was really interested in like understanding human reproduction. So trying to figure out, like how are our bodies working and in terms of reproduction. And so I used to read books on that.
Interesting. So we've got our next question here from Clyde Irwin, Elementary Magnet School. When you first started studying microbiology, were you grossed out? And if so, how did you get over that?
No, actually, I wasn't really that grossed out by it. I think mostly because we weren't working with pathogens, I didn't have to worry about it.
Sure.
And even when I started working with like really contaminated stuff, I didn't worry about it. Um, it was funny because I never worried about it. And then as soon as I got pregnant with my daughter, I started wearing gloves all the time in the
Oh,
lab.
wow. I bet, yeah.
And a bunch of the students that were working with me knew immediately, even before I told them that I was pregnant, they knew something had changed because I was being a little bit more careful.
That was the tell. There
That
you
was
go.
the tell. So, no, I don't worry about it. I mean, there are definitely some nasty chemicals, but if you use your brain and you read the instructions and really think about how to keep yourself safe, it's not a risk. So I feel very safe working in the lab and with my organisms.
It's good to know. We've got time for 2 more questions here in this segment before we have to move on to our polls, which are coming up next. These 2 questions are from Rachel and Addison. What projects in school, and I think she's talking about middle school, high school age range here, got you interested in your job? And Addison asks, did you get good grades in science when you were in school, when you were their age?
Well, did I get good grades? Yes, in high school, middle school, I got good grades in science. But it's funny, my second year of middle school, I had a biology teacher who recently retired, and I spoke at her retirement party because I didn't like her in middle school. I didn't like biology. And she was really laughing because she didn't really like me either, and I didn't really want to work that hard. And so I had to learn pretty quickly once I got to high school and college how to really, work hard at the science. But I remember my first year of middle school, I helped my teacher build a special apparatus to actually look at minerals. So, sometimes minerals like rocks will glow under UV light. And I remember helping to kind of build something in order to view that more easily. And so I think kind of in middle school, my classes on earth science just really got me really interested in how I could use my hands to solve problems. And then every teacher from then on really helped with that. Even my 7th grade class, where my teacher said I wasn't very interested, I still really liked. We did a frog dissection. She had lots of fish in her lab, and just kind of like seeing how the teacher was learning how to solve problems, and those were the things I liked to do.
So I'm curious, though, if you didn't like that teacher and that teacher didn't like you, how did you guys reconnect so that you
Oh,
were there to speak at her event?
my mom and dad are good friends with her, so I
There you
worked
go.
with her in the middle school, and it was really fun to be able to talk about how, we didn't really like each other then, but she obviously had a really positive effect on me with all of her adventures, and here I am to be able to say that it worked.
Clearly.
Yeah.
We are going to switch gears here because, speaking of fun, it's time to go to our polls. Denise has been answering a lot of your questions here, but now it's time for you to answer some that she has come up with. So let's take a look at our first poll question. And it is, when Denise was 15 years old, what did she think she would be when she grew up? Is it A, scientist, B, artist, C, teacher, or D, all of the above? I actually don't think I gave folks out there the all of the above option to vote on, so I guess we can rule that one out. But I'm able to see their responses coming in, and so far it's 50-50, scientist and artist. I'm trying to think back to our conversation here and see if you gave this away at any point. I know you talked about liking science.
Hmm.
I think there might have been some clues, though, in your questionnaire. If folks had read that before the event, might tip them one direction or other. I actually don't know the answer here. We're going to find out together. So right now, 67% think scientist, 22% now think artist, and the rest think teacher. Denise, what's the right answer here?
I actually thought I would be an artist when I grew up.
No kidding.
Yeah, yeah. I studied art. I think my parents first put me in art classes when I was in elementary school, And I even won an art contest when I was in second grade. And through high school, I took lots and lots of art classes at my high school, and I really thought that's what I was going to do. And I switched pretty early on once I got to college because I realized I could take my artistic skills and apply them to being a good scientist, to answering questions and being creative. And I actually think that that's one of my strengths. I don't think about things in a very linear manner. I think of them in a much more complicated way and really use my creative energy to solve scientific problems.
Multi-talented. So do you still do any art on
Yes.
the
So
side?
my newest art is I actually do pottery. So I've been learning how to like make things on a pottery wheel. So I'm getting better.
All right, it's time to jump to our second poll question. What completely unscientific job did Denise have in the past? Was she A, a baker, B, a kite flyer, C, a truck driver, or D, all of the above? And once again, we can rule out all of the above because I didn't add it to the interactive thing here. So we definitely have not talked about this during the live event, but I know it is in your questionnaire responses. And I am really intrigued to find out the answer, or hear you explain the answer to this. And taking a look at the responses, about half think that you were a baker at one point, 13% think you were a kite flyer, and about 40% think truck driver. And those responses seem to be holding. So, Denise, why don't you tell us what the right answer is here?
Well, I was actually a kite flyer for about 5 years.
So,
So I
what
had a summer job in high school to college where I was flying kites. So we would fly stunt kites, which you could do, or trick kites, and we would fly really, really big kites that were like 252 square feet of kite material. We'd fly those. We'd have to anchor them to, buried anchors, and you could fly different things off the kite line as a aerial display. And so I did that in Ocean City, Maryland, for about 5 years over the summer.
So, I mean, kites are really cool, and that sounds like an amazing thing to do. But how do you get paid for this?
Well, I worked at a store called the Kite Loft, and so we would
Ah,
fly displays in order to advertise the store and then give lessons on how to fly stunt kites and things like that.
that sounds pretty cool. I don't know if I would have moved on from kite flyer.
Yeah, it was a
Yeah.
hard decision. It was
Okay, let's
fun.
go to our third poll question. What countries has Denise visited or lived in as part of her job as a scientist? A, Germany, B, Belize, C, Czech Republic, D, Switzerland, or E, all of the above. In this case, I actually did remember to include the all of the above option. So that is something that you can vote on out there if you feel like that is the right answer. Um, so far, they're really quick to respond to these poll questions, Denise. So far, we've got a couple votes for Belize. Actually, votes for everything. All of the above is in the lead at the moment with about 30%. Actually, now tied with Belize. The answers are really all over the map, pretty evenly distributed. Folks are continuing to answer. Belize has taken a slight lead. Why don't you tell us what the right answer is here, Denise?
Well, I did mention Belize earlier,
You did.
but actually, E, all of the above is the right answer.
All of the above. So this was for personal reasons, for work reasons.
So I had a position for 4 years as a postdoctoral research fellow in Germany. So about 2 years ago is when I moved back to the United States. And so a postdoctoral research fellowship is the position you take after your PhD and before you get a permanent job, which is what I have now, or as a permanent research position. I did field work in Belize, so I went there 4 times. And I also did field work in the Czech Republic and visited there for conferences. And I also went to Switzerland for conferences. So I've presented my research in those countries and also in Germany too.
Do you speak German from that?
A little bit,
Little
yeah.
bit?
So I speak German, but I'm not completely fluent.
And you know enough German to be a big fan of German hip hop?
Yes, I do. I'm a big fan of German hip hop,
I
actually.
had, I have to admit, I
That's
hadn't
what my
listened.
daughter was raised on, was German hip hop.
I'm sure she will take that with her for the rest of her life. I have never really listened until this morning when I was reading through your questionnaire again to the German hip hop, and I might be a new convert as well. It seemed pretty cool. All right, we're going to go into poll number 4 here. Bacteria are often thought of as the bad guys of nature because some can cause illness. Which of the following are beneficial activities of bacteria? Is it A, eating toxic waste and pollutants? Is it B, nutrient cycling in soils? Is it C, making methane? Is it D, oxygen production? Or is it E, all of the above? Which are beneficial activities of bacteria? Folks are entering in their answers out there, Denise.
Okay.
Once again,
Let's
very
see if they
quickly,
remember.
40% at the moment think all of the above. Eating toxic waste is getting some votes. Nutrient cycling getting some votes. Oxygen production also getting some votes. Nobody so far has voted for making methane. And as the voting settles out here, 63% think it's all of the above. Denise, are they right?
They are right. All the bacteria can do all of these things.
Okay. We're going to move along now. We're going to talk about the future, Denise, for a moment here. And we've had some questions coming in related to both your future and our students' futures. A lot of folks out there, let me pop these questions up, are wondering if you weren't doing what you do now. I think we're going to have to scroll down a bit to see those questions that come in. What would you do if you were not doing your job now? So Austin and Rachel want to know, if you had to get a new job unrelated to science, what would it be, and what would your second choice of job be?
Well, my second choice of jobs, when I interviewed to move back to the United States, I applied for my research position at the U.S. Geological Survey, and I also applied for positions as a professor at different universities. And it was actually a tough decision of where to go, but I really like focusing 100% of my time on research. So if I wasn't here, I would be at a university and I would be a professor there. I really haven't thought about what I would do if I wasn't in science. I really enjoy teaching, but I would want to teach science and I'd want to, be a professor. So I think sometimes it'd be fun to go and be an artist again, or maybe be a cook or a gardener, but that's a tough one. I like what I do too much to really think about changing.
It's a good problem to have, not knowing what your
Yeah.
next career would be. It means you're very happy in your current one. Our next question is from Addison, and she asks, if you have an experiment dream, how do you make it come true?
Experiment dream. Hmm. Well, I do dream about experiments, Addison, so that's not an unusual thing. Um, Sometimes it's a lot harder to make the things you imagine happen than you think it's going to be. And so, usually, if I think up a new way to do an experiment or a new way to look at microbes that I don't know how to do myself, I'll find collaborators that can help me. So, I'll work with other scientists. I have a great research associate in my lab who works with me. And we often can, if I come up with a crazy idea in the middle of the night, we can usually hash out a really good plan the next day on how to make that happen. And so I often will, I'll look online, I'll do some research in the literature to see if anyone else has had a similar idea, and then I'll talk to my fellow scientists and get ideas from them.
Very cool. Well, we were just, during the poll segment, talking about all of the places in the world that you have been able to travel for your work. Our next question is related to that. It's from Clyde Irwin Elementary Magnet School. Are there places in the world you would want to go to do further research in microbiology?
Definitely. The list is very long because I want to go to some really cool places. Right now on the list, I'm hoping to get some research funded that we could go to Alaska and do some work there. I would love to go to Antarctica or somewhere in the far north. I've had colleagues do research there. Right now, the biggest limitation is figuring out what kind of scientific questions I can ask there. And so those are things that I have to figure out. Trying to find good scientific questions I can ask in really, really unique places. And but I really want to see the whole world, and being a scientist is a great way to do that because it gives me a lot of freedom to travel as long as I have interesting conferences and places that I can go present what I'm doing. And if I have interesting questions that can be applied to unique places.
We've got another question from Yena. She's asking, have you ever been under sea? Have you gone under the ocean to look for microbes?
I have not. I'm thinking about this. When I did a lot of the marine biology work where I was scuba diving, it was actually before I became a microbiologist. And so I've never actually been out in the oceans to look for microbes. I have helped people in coastal environments do field work to look for microorganisms, but a lot of that work was done from the boat and coring from kind of the side of the boat. So I've never been undersea yet. I would love to go down to the hydrothermal vents and see those and look for microbes there, but hasn't happened yet.
Okay, we've got a great question next from Mrs. Diamond's fifth grade class. Is there a way to replace fossil fuels with microbes?
There's a lot of research right now looking at biofuels, and so that is using like algae and specifically algae. So those are a type of uh, eukaryotic microorganisms. So those are algae. They're a little bit different than the bacteria that I normally study. But there's a lot of work being done to find out if those organisms could be grown to harvest the oil that they make as a byproduct of their metabolism and use that for biofuels. I'm not involved in that work, but there's some really interesting things going on where biofuels are being thought to are a good target for replacing fossil fuels.
And actually, I believe our next role model is involved in that, so we're going to talk about him in just a moment, but we have time for just one last question here. This one is from Yena again. Have you discovered any new types of ways that microbes help humans?
I'm actually in the process of trying to find that out right now. So many, you may have heard in the news of something called hydraulic fracturing or fracking. It's a new way to get natural gas out of the ground or even oil out of the ground. And there's a lot of concerns about how safe it is. So one of the things I'm trying to find out is whether or not microbes could be used to protect the environment or to degrade any contaminant, protect the environment by degrading contaminants that might occur due to that process. And so one of the things that I'm really trying in my research is to find out how we can harvest energy in the best way and how we can, have the best practice for actually doing that. So I'm hoping to evaluate that in the next years or next few years. So
Very cool. So hopefully we'll be able to connect with you again in a couple years and see how that research is going. It sounds really interesting.
it's really cool. I like it
So,
a lot.
unfortunately, we're all out of time. This has been really fascinating to learn more about microbes and all of the interesting things they do and the work that you do and your career path. This has really been an education for all of us today. So thank you so much for taking the time and sharing the story of your STEM career with us.
Well, thank you so much. I really enjoyed taking the time and talking to you all.
Okay. As I mentioned before, our next STEM role model also involved in biotechnology. His name is Larry Feinberg. Larry actually started out as a high school science and Spanish teacher, but today he has a PhD in microbiology, and he is a CEO of a biotech startup company that he founded to help solve some of the world's biggest sustainability issues. We're going to learn about Larry's work and his STEM career path on May 29th at 1:30 p.m. Eastern. Once again, that's all the time we have. I'm Patrick Shea. Thanks for joining us on Jason Live. We'll see you next time.